On today's GARNet Research Roundup, I'm really delighted to be joined by Professor Ari Sadanandom from Durham University. And we're going to be talking about a paper just published in the plant Cell, the title of which is SUMO Suppresses the Activity of the Jasmonic Acid Receptor CORONATINE Insensitive 1. So thanks very much, Ari, for joining us today. And it'd be great if you give us an overview of this paper, which really brings in lots of different aspects of plant biology.
Yeah, so it took us a while to get involved in this. It's only part of the picture, but what we tried to do in that paper is try to figure out how we had this mutant called overly tolerant to salt 1 and 2, so it's OTS. Mm-hmm. These mutants have reduced root growth in salt, but they also had a disease phenotype where they have increased salicylic acid, but also increased defense against biotrophic pathogens like Pseudomonas syringae, but has got enhanced, it's more susceptible to botrytis, which is more of an ectrophic pathogen.
Okay.
So we wanted to try and figure out what does that really mean. So then we started to go down this angle of whether jasmonic acid could be involved. So when we put those mutants on jasmonic acid, it was quite, the OTS mutant is quite resistant to jasmonic acid. In other words, when you put plants on jasmonic acid, seedlings on jasmonic acid plates, the root length is reduced. Mm-hmm. So that's one way to see the sensitivity of the mutant to jasmonic acid. But the OTS mutant is more resistant. Okay. So it seems like it's more resistant to jasmonic acid. In other words, it's not being able to sense jasmonic acid, or the signaling is blocked, one of the two. So we then tested whether the sensitivity is the same or not. It turns out to be the same, but then we found out that the signaling is repressed. So you can perceive it, but you can't signal.
So these OTS mutants from your previous work are involved with SUMO signaling, and then you managed to. So what you did then is link the SUMO signaling with this JA response, and this is the key aspect of the paper. Yeah.
That's right. So the original mutants were overly tolerant to salt. So they were salt sensitive mutants. Then they've now linked that to jasmonic acid through sensitivity to necrotrophic pathogens like botrytis, grey mold.
Okay.
So and we found out that because they're not signaling for jasmonic acid, there must be some block in the system, and that block turns out to be the JAS repressors who repress JA signaling.
Okay.
And they are simulated. And we did mass spectrometry, figured out what the simulation site was. And once they're simulated, what happens was these things are JAS, but they're simulated. How are they actually blocking JA signaling? Mm-hmm. One thing what we found out was the JAS repressors are accumulating, and there are two types of JAS repressors. One is the simulated form, and the other one is the non-simulated form. And they both accumulate in the OTS mutants.
Okay.
And how does so this correlation, why does the simulated JAS go up, and how does it enhance stability of non-simulated JAS? Mm-hmm. And the only way the kind of major way how that could happen is by transcription of gene expression for JASs. That was more or less the same. So the SUMO E3 ligase, for example, was expressed at the same level, so there's no increase in ligase, adding the SUMO on. But what we found out the other way is that you can then block the degradation of JA.
Okay.
And the only way to block degradation of JA, so far we know, is that the COI1 JA receptor binds to jasmonic acid, and then it acts as ubiquitin E3 ligase to degrade the JAS repressors, and thereby trigger JA signaling.
Okay. Okay.
And we found out that the simulated JAS proteins binds to the COI1 JAS receptor, JA receptor, mm-hmm. and prevents it binding to the non-simulated JAS.
Okay.
And because it can't bind to the non-simulated JAS are stabilized, and they block jasmonic acid signaling. And this occurs in the OTS mutants.
Okay.
And that's how we found the link from salt to simulation of JAS proteins, and how mechanistically the simulated JAS proteins can attenuate the degradation of JAS repressors, and therefore you block JA signaling.
Okay, that's really interesting that you might imagine if you found that the level of simulated JAS was up, the level of unsimulated might be declined, but you found that they're all at the same level.
Yeah.
So do you see, so you said that the simulated Jas proteins somehow like block the COI1 receptor. Do you, well, within the paper, you found something about the specificity of that response. So can you say something about the molecular interaction that occurs there?
So when proteins are simulated, they can do one of the things. The simulated SUMO is conjugated onto target proteins, and that can block interaction with this normal interacting partner.
Yeah.
And the other way is that the simulated target protein can interact with other proteins which have a SUMO interacting motif or a SIM.
Okay.
And we found this motif in the COI1 receptor. So simulated Jas binds the COI1 receptor, okay, and this it's the SIM motif in COI1.
Okay.
That's the specificity. And not only that, is that this binding, so COI1 needs to be bound to jasmonic acid before it binds to the Jas repressors to degrade it. Okay. But the simulated Jas binding to COI1 is independent of whether you have jasmonic acid or not.
Okay.
So this is unique. So
Okay.
this has not been seen before. So whether you have jasmonic acid in the cell or not, if you trigger the SUMO system to simulate Jas protein, it'll bind to COI1 and it will repress signaling of Jas, JA, signaling of jasmonic acid, you see.
Okay.
And this is, we said in the paper, this is because plants have a two-pronged approach when their stress comes. One is the fast approach, and the other one is more long-term adaptive response. So the fast adaptive response is imagine like simulated Jas accumulates and it blocks COI1 from degrading Jas, and Jas both accumulate and repress JA signaling. And that needs to happen whether there's endogenous Jas, JA jasmonic acid is around or not, because you don't have time to degrade them and reset the system.
Yeah, I see.
You see, so that's where the system comes from.
So do you have any evidence for how long this interaction might happen? Is there any way, do you think, to find that out, or is that something you might want to look at in the future?
Yeah, that's something that we're trying to work on also. How quickly does simulated Jas accumulate? We know within 10 minutes it goes up.
Okay.
So talking about in vivo, it could be seconds, right? But remember, if you do Western blot, you pull out protein, all this takes time. But in vivo, it could be seconds. So if we have a reporter for simulated Jas, we may be able to pinpoint which cell types this occurs and also how fast this occurs.
Okay.
That's the next step, really.
Okay. And also, so as I said, the paper is really nice. I enjoy this sort of paper which has this stepwise look through. So you can start with a botrytis infection. You go through simulation, which then affects the JA response, which ultimately then affects the plant's response back to the botrytis, and the botrytis tries to mitigate this response by changing the JA response. But this seems to be, all this work is done in Arabidopsis, but this seems to have a really, potential application, in the field. So is this something you're trying to move into other plants, or are you looking elsewhere for an?
That's right. We're looking at JA responses in wheat against another necrotrope called Septorium. So number one disease in wheat at the moment, and all the fungicides are breaking down against Septorium.
Okay.
So if you can modulate JA response in wheat by either using a chemistry that blocks the SIM on COI1, and therefore COI1 is then released to degrade Jas repressors, you can imagine you can switch on JA response in wheat, and that should give you protection against Septorium.
Yeah, I see.
So these kind of applications you can see, because we can identify a specific motif in the JA receptor, you could then make a chemical, or you can modify that motif, then you get a gain of function.
Okay. I could see that you could use, CRISPR gene editing to modify that motif in the wheat COI1 and see what you get. Yeah, definitely.
Yeah.
Okay, cool.
That's right.
All right, excellent. So thanks very much, Ari, for talking to us about the paper. And as I say, encourage people to have a look through. It's a really nice progression through these different parts. So thanks very much for chatting to us today.
No worries. Thank you.